Ever wondered how identical twins with the same genetic makeup can have different health outcomes? Or how a disease like cancer can manifest so differently in individuals with similar lifestyles? The answer lies in the fascinating world of epigenetics. Epigenetics, a term coined by Conrad Waddington in the 1940s, is the study of changes in organisms caused by modification of gene expression rather than alteration of the genetic code itself. Its name is derived from the Greek word epi, meaning on top of, and genetics, implying it's a layer above the conventional genetic makeup that we inherit. The journey of understanding epigenetics in cancer has been a winding road. In the early days, cancer was primarily seen as a genetic disease caused by mutations in the DNA sequence. However, as research progressed, scientists began to notice a pattern. Certain genes, known as tumor suppressor genes, were being switched off in cancer cells despite having no mutations. This observation led to the realization that something beyond the genetic code was at play. In the 1970s, researchers discovered that chemicals could attach to DNA and modify its activity without changing the genetic code. These chemicals, known as methyl groups, were found to be abundant in cancer cells. This process is known as DNA methylation, one of the key mechanisms of epigenetic regulation. Further research in the 80s and 90s highlighted the role of histones, the proteins around which DNA is wound. Changes in these proteins, known as histone modifications, were also found to influence gene expression, adding another layer to the epigenetic regulation of cancer. Fast forward to the 21st century, and the field of epigenetics has exploded. Advanced techniques such as next-generation sequencing have allowed researchers to map the entire epigenome of a cell, providing unprecedented insights into the role of epigenetics in cancer. Today, researchers are focusing on how to harness these insights for cancer prevention and treatment. Epigenetic drugs designed to reverse abnormal DNA methylation or histone modifications are showing promise in clinical trials. In addition, understanding the epigenetic changes that occur in cancer can help in early detection and prediction of treatment response. In conclusion, the story of epigenetics in cancer is a tale of scientific discovery, from the first observations of gene silencing through the unraveling of DNA methylation and histone modifications to the cutting-edge research of today. Epigenetics has transformed our understanding of cancer, revealing it as a disease not just of genetic mutations but of dysregulated gene expression. As we delve deeper into this fascinating field, we are not only unraveling the mysteries of life, but also opening up new avenues for cancer diagnosis, prevention, and treatment. Who knows what exciting discoveries the future of epigenetics in cancer holds?